The stage is yours.
Thank you. Hi, everyone. I'd first like to thank the organizers and the committee for giving me the opportunity to present my work to you. My name is Inbal Benhar. I'm going to present work I did as a postdoc in the lab of Aviv Regev at the Broad Institute. And I would first like to acknowledge my colleagues and collaborators, members of Aviv Regev's lab, Josh Sain's lab at Harvard, and Jagan He's lab at Boston Children's Hospital. So the challenge I set out to address is that of CNS regeneration failure, central nervous system regeneration failure, in light of multidimensional tissue complexity. As most of you probably know, neurons in the adult mammalian central nervous system generally fail to regenerate after sustaining injury or disease, and this leads to irreversible and often devastating loss of function. Now, this challenge is even greater when we consider just how complex the CNS tissue ecosystem is, both at baseline and more so after injury or disease, with dynamic changes across time and space in cell composition and cell states, including neurons, glia, immune cells, and more. And besides these changes in time and space, we also have a lot of interactions across the different cell types in the tissue. So while much insight has been gleaned from studying each of these components in isolation over the years, we can now graduate towards multicellular understanding, and we finally have the tools to allow us to do that, to hopefully gain better insight into what happens and what goes wrong in CNS pathology. The model we've been working with is that of optic nerve crush injury in the mouse. This is a well-established model of CNS degeneration and regeneration, in which crushing the axons of the optic nerve results in extensive death of retinal ganglion cells, these neurons in the inner retina. Within 2 weeks, about 80% of these cells are lost. You can also appreciate this here in this whole mount staining of a mouse retina with a retinal ganglion cell specific marker. However, it's important to note that retinal ganglion cells are not by any means a homogeneous population. We recently showed that there are as many as 45 molecular subtypes of retinal ganglion cells. They differ by many additional measures as well. And more importantly to this context, these subtypes also vary dramatically in their ability to withstand neuronal injury, with some surviving far better at this 2-week time point than others. So we have resilient subtypes and susceptible subtypes. And this of course prompts a question as to what drives this phenotype and how we can use it to hopefully rescue more neurons. As a neuroimmunologist, I always consider the fact that neurons do not live in isolation, but rather there is a whole cohort of cells participating in the injury response. And in this cartoon depiction of the retina, you can appreciate some of these cells, including resident glia, such as Mueller glia and astrocytes, the CNS resident macrophages, microglia, epithelial cells, and others. So of course, after injury, these cells all undergo dramatic changes. And on top of that, we have infiltration of immune cells that come in from the periphery, from the blood. So now switching this over and looking at the same cells in gene expression space, this is an overview of about 150,000 immune glia and epithelial cells that we profiled using single cell RNA sequencing from mouse retina at baseline and at 6 time points following optic nerve crush out to the 2-week time point when retinal ganglion cell death plateaus. So each dot on this map represents a single cell, and cells that are clustered together and colored by the same color are transcriptionally similar, and so they represent cell types or states. Now that we have this elaborate atlas, we can start asking questions on the cascade and the dynamics between the different cell types. And I'll show you some of the examples of what we found. So the earliest signals that we found in the tissue after injury were of resident glial reactivation. This includes astrocytes and Mueller glia. And in astrocytes, this activation response also included the upregulation of genes encoding chemokines that call in neutrophils and monocytes into the tissue, which is actually the next event in line. So you can see here by FISH, expression of CCL2, a monocyte chemoattractant, by astrocytes in the inner retina at this 12-hour time point. So it looks like this activation signal early after injury works to facilitate the ensuing immune response. Now we will focus on this immune response, and when we order the cells, the immune cells in our data, based on their maximal frequency throughout the time course, we end up with a cascade that is very much reminiscent of the wound healing cascade seen in other parts of the body. We have early infiltration of neutrophils and monocytes, followed in the intermediate time points by the expansion of macrophages, and in our case, in the CNS, also proliferation of microglia, resident macrophages. At the later time points, we have expansion of adaptive immune cell types. So when you put it all together, you have all the cellular components of what you would see for a skin injury for example, except that in the CNS, this elaborate cascade is insufficient to restore functionality and to create sufficient repair. We focused further on monocytes and macrophages, which were the most diverse in our data set. So I showed you that monocytes come in pretty early, and if we look at it at the tissue with transgenic mice, you can see monocytes in red and yellow, and you can appreciate that they're virtually absent from control retina, but they rapidly infiltrate early after injury, starting with this crown around the optic nerve and gradually spreading peripherally across the ganglion cell layer. So they're really in the right place and at the right time. When we zoom in closer, we see that these monocytes differentiate and acquire macrophage morphology, and they come in intimate contacts with retinal ganglion cells and their axons here in green. So it's very interesting to find out what the fate of these cells is in the tissue. As I told you, macrophage subsets expand later on in the time course, and I'm going to focus specifically on two subsets that we found very interesting that differed mostly by MHC expression levels. When we looked closely, we saw that these subsets are also present in the uninjured retina in the steady state, although at lower levels. And as you can hopefully appreciate here, these MHC high and low cells occupy distinct spatial locations in the retina at the steady state. However, after injury, they both expanded in numbers and they intermingled. The spatial segregation was much less defined. Among the subsets that we found were some novel ones, but also existing subsets that people had described before, but didn't have the tools with which to access them and profile them better. So we now have the tools with which to interrogate these cells and target them in the tissue to understand further what it is that they do in the healthy state as well as after perturbation. Finally, we wondered whether this early monocyte infiltration and the later macrophage expansion are connected, or in other words, whether monocytes could be the source of these macrophages. To this extent, we used an anti-CCR2 antibody, which effectively depletes monocytes in the blood, and as you can see here, also depletes them in the retina. And we collected immune cells and subjected them to single-cell RNA sequencing. This is, of course, from injured mice in both cases. And we see that monocyte depletion results in a decrease in these macrophage subsets. So this indicates that in the context of injury, at least to some extent, monocytes do contribute to the expansion of these macrophage subsets. The time I have today allowed me to show you some of these discrete events that occur within certain subsets, but I told you at the beginning that I'm actually much more interested to look at how all these processes are co-regulated across the tissue and how the tissue works as a complex unit to govern the injury response. So one way in which we try to approach that is using the dialogue method that was developed in the lab. This is a computational method that uncovers co-regulated transcriptional programs across different cell types. One such co-regulated program was upregulated in our data set at the 4-day and 1-week time point. And it was characterized by high expression of interferon response genes. We validated that at the protein level as well. You can see the IFIT3 washing over the tissue. And this was very interesting to us since interferon and the interferon response has different effects in the CNS based on the timing and the dosage at which it is present. But more specifically, when we look at the time points here, if you remember the histogram that I showed you in the beginning of retinal ganglion cell death, this correlates tightly to the time point at which their numbers are greatly dropping. So now that we have this, this very prominent signature of the other cells, we're curious to see how it is relevant in the context of this injury. So putting this all together, what I showed you today is a molecular atlas that we created of the non-neuronal retinal cells and their changes after traumatic injury. We looked both at cell type specific programs such as reactivation and proliferation, and we graduated towards coordinated programs across cellular compartments. For example, I showed you the interferon program. We also gained new insights into retinal macrophage heterogeneity, both at the steady state and after injury. And we're very excited to have all these tools with which to follow up on the many questions that have arisen as a result of finding out these subsets. Altogether, we can say that CNS injury induces an orchestrated response across the tissue where various cell types work in a coordinated manner to at least try to resolve damage. Apparently, they do that insufficiently. By identifying the key players and events, we can hopefully understand why the response falls short or how it is selected towards certain neuronal types. And that would hopefully help us highlight evidence for intervention. And as a final note, I think that we're in a very exciting place where we're now poised. We have the computational and the technological tools to look at tissues at scale. And as someone who's very much interested in tissue biology and tissue repair, I think this paves the way for us to understand tissues across the body, within the CNS and out of it, much deeper, both at the steady state in health and in disease. So again, I would like to thank my partners in this endeavor, and I thank you all for your attention, and I'm happy to take questions.
Thank you very much, Inbal, for this great talk. We have questions from our panelists. Let's start with Stefan Jung, please. Yeah, you can ask Stefan. We don't hear you. Maybe Ethan Gross, if you want to start, and then we go back to Stefan.
So can I see the see who talked, who spoke?
Yeah, Inbal, maybe you can remove the sharing.
Yes, Inbal.
I'm sorry. Okay.
Thank you, Inbal, for
Hi.
a very interesting talk. So what is your vision? What do you think is happening at the end of the road? So you have monocytes, you have macrophages. Let me know. I have certain vision, and we'll see if it's the same vision that I have. But
what do I think is happening?
At the end of the road, what are they trying to do, these cell types?
These cell types are trying to do what I believe immune cells try to do wherever they go, which is fix damage and repair the tissue. So they come in, they bring in the machinery to clean up dead cells and debris. They bring in first pro-inflammatory factors, then neurotrophic factors. But it seems that whatever they do is insufficient. We know from our earlier study that retinal ganglion cells have very strong cell intrinsic signals that prevent them from regenerating. And a lot of studies work on overcoming those intrinsic signals. But we also know that it looks like combinatory therapy is the way to go. In short, yeah.
Inbal, and it's not a crazy idea, but it's something that people see. Is it possible that they're also trying to trigger de-differentiation of glia cells to replace the damaged cells, not only cleaning up the damage, but generating cells from nothing or from...
That's a great question. So I would say I wish, and we do see that in different animals. We see that in the chick retina and in the zebrafish retina. And there was actually a really nice paper that came out about a year ago showing Mueller glia have the capacity to de-differentiate and replenish other retinal cells. But it looks like if you compare chick, zebrafish, and mouse, they all start after injury. They have this transcriptional state that they revert to. They go into gliosis, but then the Mueller glia in the mouse stop and go back to what they were, as opposed to the other two that managed to de-differentiate and complete this process. So something went wrong in evolution or went right, I don't know, but we still have a way to go in terms of trying to understand. But definitely this stem cell or progenitor avenue is something that these are all things that are connected. We just see it to a very limited degree in the.
So what do you think is the relevance to neurodegenerative diseases?
So I think that what we see in neurodegeneration is very similar. We just have longer time scales. We also see many examples of neuronal specific resilience in Parkinson's disease and Huntington's, where you so-called have the same insult, whether it's genetic or otherwise, that impacts all the neurons, but yet some of them are more vulnerable and they die and others do not. So we have, these themes that recur in neurodegeneration and in acute injury. And I think we can definitely learn from these acute models and hopefully transfer it.
Wonderful.
I think the tricky part, thank you very much.
Go ahead, sure.
The tricky part is that in neurodegeneration, we often realize far too late. And then that's a question of when and how to interfere.
Wonderful, good luck.
Thank you.
Thank you. We move from one more question from Michal Schwartz.
I think Stefan tried before, but
Stefan, yeah,
but I will be more than happy to ask. So thank you, Inbal. Beautiful talk.
Thank you, Michal.
Yeah. So my question to you, when you depleted the monocytes by using CCR2, did you try to see whether the phenotype of the microglia is affected by the monocytes?
Yeah, so we did that to some extent. There were some computational issues with that specific question, but I do see that the interferon subset of microglia seems to expand as a result of the monocyte depletion.
Very nice.
Which I know that is, in line with some of your recent
also
results.
It was recently reported with respect to the COVID-19 in a work that was published by the group in Stanford that they found in post-mortem patients that die out of COVID, there is this very strong signature in the choroid plexus of type 1 interference. So very nice.
Thank you.
Okay, thank you very much, Inbal, and thank you for